So our next speaker is David Hibbett from the Department of Biology at Clark University in Worcester, Massachusetts. David's a leader in the field of fungal evolutionary biology, and the unifying theme of his research is deciphering the phylogenetic relationships between species. He has a special interest in Basidiomycetes. This is the group of fungi that includes mushrooms and a lot of fungi that degrade Lignocellulose, which is, of course, of special interest to us here at the JGI. One of the perils of modern academic life is that your students can publicly rate you on websites. And so I didn't know David, but I wanted to find out what we could expect. So I checked ratemyprofessor.com. And his students really like him. He got very good marks. So I think we'll have a real treat. But there were some selected comments I wanted to relay to you. Surprisingly not boring for a mycologist. He's funny, but not at all annoying in his humor. And then my favorite is professors like Hibbett are rare. Too bad he studies mushrooms for a living. Which kind of almost has like a haiku-like simplicity to it. He also got a coveted chili pepper next to his name for hotness. I'm not exactly sure what that means. I got that once, and my wife got really annoyed with me. So David's going to talk to us this morning about evolutionary perspectives on the diversity of lignocellulose decay mechanisms in Basidiomycetes.
Okay, thanks a lot, Dan. I didn't know you were going to go to Rate My Professor. Okay, it's a real honor to be here. It's a great opportunity to come and speak to a crowd that I don't usually get to talk to, so my thanks to the organizers for giving me this opportunity. I realized as soon as I had submitted the title for this talk that it was a really dry and boring title. And so I have this alternative title here, How Mushrooms Changed the World, which I hope you'll forgive me. That might be considered kind of a Yags-inducing title. Nonetheless, I hope to convince you by the end of the talk that mushrooms have changed the world. So I'm a fungal systematist, and the group of fungi that I'm most interested in are the Agaricomycetes. And if that name is not familiar, the organisms are. These are the mushroom-forming fungi, so the vast majority of the macrofungi you see out in the woods are members of this group. There's about 21,000 described species, which is about a fifth of all the known fungi, and God knows how many are actually out there in nature. So a large and important clade of fungi, and they're beautiful. I'm interested in the morphology and morphological evolution, but today what I'm going to talk about is their ecological evolution and the evolution in their nutritional modes, the way they're making a living out there in nature. And these things do really important things out in nature. The major nutritional modes for the Agaricomycetes are saprotrophy, so decayers of dead organic matter. There are litter decayers, and then also wood decayers, and that's the major focus today. This is the group of organisms that contains the major concentration of wood decay microbes, so very important in the ecosystem. There are also ectomycorrhizal fungi in this group, so the vast majority of ectomycorrhizae are here, although there are also ectomycorrhizal ascomycetes. Some plant pathogens, not a whole lot, compared to other groups anyway. And then some weird things like basidiolichens, mycoparasitic fungi, insect symbionts. So they're very flexible in how they make a living in nature. But the focus today is on the wood decayers. And wood, as you all know, is very important stuff. It's a very tough substrate. It's a major pool of organic carbon in terrestrial ecosystems. It's resistant to microbial attack, which is why we make buildings and things out of it. And wood is a complex of carbohydrates, which provide carbon nutrition for fungi, like cellulose and hemicelluloses. But also, wood is largely composed of lignin. And Jody Banks had a great introduction to this stuff this morning. So lignin is a major component of wood cell walls. It's a recalcitrant, heterogeneous polymer. It's a barrier to microbial attack of the carbohydrate portion of the wood. It's not a significant carbon source for fungi. And what's important, and what I'll emphasize today, is that it's a major precursor of coal. Okay, so that will come back in just a moment. Agaricomycetes have evolved two very different ways of decomposing wood. White rot, which I'm sure most of you here have heard about, and also brown rot. White rot is kind of a brute force approach to wood decay. In white rot, all the components of the wood cell walls are decomposed, and the substrate is left bleached and stringy after colonization. Enzymes called class 2 peroxidases are very important in lignin removal in white rot. So things like manganese peroxidases and lignin peroxidases and versatile peroxidases are in this group of white rot fungi. Brown rot is a somewhat more mysterious mode of decay in which lignin is modified, but it's not appreciably degraded. And after colonization by the fungus, the substrate has this brown, cubical, sort of crunchy, friable consistency. And one of the distinguishing features of brown rot is that depolymerization of cellulose has some non-enzymatic mechanism for at least the initial depolymerization. So these fungi have evolved some way of pulling out the carbohydrates while leaving the lignin behind. So it's a very elegant mode of wood decay. And there have been numerous transitions between brown and white rot in the Agaricomycete fungi, including within individual groups. So for example, the polyporales, the polypores, a group I'm particularly interested in, there are white rot and brown rot fungi intermixed. And you can't tell if a fungus is white rot or brown rot by looking at the mushrooms. You have to look at the substrate. Okay, so Agaricomycetes and fungi in general play tremendously important roles in ecosystems. They're really important in forest health and in carbon cycling. And they've been important for a very long time. You know, we have fungal fossils from the Rhynie chert, so we know that they've been central in terrestrial ecosystems as long as there have been at least eukaryotic terrestrial ecosystems. And yet, when you look at textbooks, when we tell people about the history of life, the fungi frequently get kind of obscured. So here's a timeline from a textbook that I use in my introductory biology class, and there's lots of animals and plants, very nice, but no mushrooms, right? No fungi. It's like they're just not there. Similarly, when you look at pictures like this, or the nice one that Jody showed of a carboniferous swamp forest earlier, you know, you might look at this and see, oh, you know, very nice, all of the organisms that you now know are lycophytes, and the obligatory large animal there. But when I look at a picture like this, I think, where are the fungi? You know, surely they were there. Surely they were important. And is this a blind spot in our thinking about the world and therefore our understanding of the world? So that's kind of the subtext for the whole talk today. Now, one person who did not ignore fungi when thinking about the history of life was a geologist named Jennifer Robinson, who wrote a really great paper in 1990 in which she was trying to understand the changes in organic carbon sequestration over geologic time. And as you all know, and as Jody explained, during the Carboniferous and into the Permian period, there was a tremendous amount of sequestration of organic carbon derived from plant material, derived from those lycophytes, and specifically from their lignin, that gave rise to vast coal reserves exceeding those formed in all other periods. And so why is that? So Robinson addressed that in her paper. Now, before I get to Robinson's specific hypothesis, let me just say a few words about coal. Coal is, you know, it's a mega commodity. You know, so what is this? 6 billion tons consumed worldwide in 2010, 45% of the electricity in the US. So it's very important stuff. So it's of interest to understand why we have coal and why at the end of the Permo-Carboniferous that organic carbon sequestration declined. So if you want to get lots of information about coal, if you want all the good news about coal, you can go to the website of the World Coal Association, as I did, where there's this informative page about what is coal. And they explain that it's a fossil fuel derived from prehistoric vegetation. All right, fine. They explain about how living plants trap solar energy. And then they go on to say, when plants die, this energy is usually released as the plants decay. Under conditions favorable to coal formation, the decaying process is interrupted. And that's, I think, probably what most people think about when they think about coal formation. You know, why did coal form? Well, the giant arborescent lycopods fell into the swamps. They got trapped in these anoxic waterlogged sediments, and decay just didn't happen. And this perspective, which I think is commonly held, it reveals kind of a bias or an assumption, which is that decay is a process that has always just happened. You know, it's like a geological process that's ongoing. And under some conditions, when decay is inhibited, well, you get peat, and then ultimately that can become coal. And there's probably quite a lot of truth to that, but Robinson also thought about the biotic factors that might have been involved in this pattern of carbon sequestration. And there were two components to her reasoning. One was her observation that Basidiomycete fossils were very rare in the Paleozoic, so this was pre-molecular clock, and then they became more abundant later on. So recognizing that Basidiomycetes are the major lignin-degrading organisms, she thought, well, maybe they were just absent during much of the Paleozoic. Another component of this, which again gets nicely back to Jodi's talk, has to do with lignin in lycophytes. And she argued that these arborescent lycophytes and other plants at the time had a heavy investment of lignin in their tissues. So there were those two elements there. So putting this all together, this was her hypothesis. She says, you know, as dominance shifted from lycopsids and pteridophytes to gymnosperms to angiosperms, plant architecture became more sparing in its use of lignin. And the important part for me is that she then goes on to say, lignin-degrading organisms were rare or absent in the Paleozoic, but diverse and abundant in the Tertiary. So the reason that you had organic carbon sequestration declining at the end of the Permo-Carboniferous was because that was when the lignin-degrading organisms came into existence. So with that introduction, then, the questions I'm going to try to address, there are two of them. One is, you know, what are the mechanisms of these transitions between white and brown rot that have occurred repeatedly in the Agaricomycetes? What are the important gene families? And when did these transitions occur? In particular, when did white rot, when did the ability to decompose lignin first make its appearance? And how might that have affected the history of carbon sequestration? And I've been working on this project through one of the projects that's part of the JGI's fungal genomics program. Our project is called the Saprotrophic Agaricomycetina Project, or SAP. And it's, we're seeking to construct 30 genomes of mostly wood decay fungi. And this is one of a number of projects in the fungal genomics program. There's one on mycorrhizae, there's one on plant pathogens, there's programs on yeasts, on schizophyllum, et cetera. So it's been a real godsend for fungal biologists. And the big one that started this year is a 1,000 fungal genomes project, which is really exciting. And I think this is going to sort of promote the catalyzation of mycology into truly a genome-driven, or at least genome-enabled science. So we're not at 1,000 fungal genomes yet, and we're not at 30 genomes in the Saprotroph project yet. We have about 20 genomes done so far. I'm going to present results using the first 12, which are shown here. And I'm going to spare you as much as possible taxonomic names because you don't care. I care. Anyway, we have 5 brown rot fungi, 6 white rot fungi, and 1 mycoparasite. And they've been selected with an eye towards not just their nutritional diversity, but their phylogenetic diversity. So it really spans a lot of the fungal tree of life. Being a systematist, of course, the first thing we did was make a phylogenetic tree. And since we have whole genomes, we have lots of data. I won't belabor the details. We have 4 different data sets, 2 different methods of analysis, so 8 total analyses. Most of the nodes in the tree get maximal support in all analyses. 6 do not. These are those nodes, and the red cells show you that in at least 4 out of the 8 analyses, we have strong support for all these nodes. So we're fairly confident in this tree. It's a hypothesis, of course, but it's a pretty robust one, we think. This is mostly Agaricomycetes, so from about this point on up, it's Agaricomycetes. The purple are some mycoparasites and a human pathogen. The brown ones are 4 different brown rot lineages. The white boxes are white rot fungi. That one lone green fungus up there is mycorrhizal, and that's sort of a soil and litter decomposer. And so you'll see this tree and these color coding in subsequent slides. So we're interested in much more than the phylogeny. Of course, we're interested in the diversity of enzymes that are involved in wood decay. It's a very complicated process. I'm not a biochemist, so I rely heavily on my collaborators for help. There are basically 3 or 4 classes of reactions that we're interested in: lignin degradation, hydrolytic degradation of carbohydrates, and oxidative degradation of carbohydrates. And then there are accessory enzymes that produce peroxide and other things necessary for these reactions. I'm going to focus on class 2 peroxidases because of their central role in lignin degradation, and I'll mention a couple of cases that are involved in degradation of crystalline cellulose as well. But this is just a small part, obviously, of the project. So there are 27 gene families that we were focusing on, 17 cases, 11 oxoreductases, white rot fungi here, brown rot fungi here, the one mycorrhizal thing here. This is a heat map, obviously, and if you kind of squint your eyes, I think you can get the picture that white rot fungi are enriched in just about all of these gene families relative to brown rot fungi. I should also mention, I should have mentioned earlier, these are a bunch of other fungi, other fungal genomes that we also accessed to conduct this project. Okay, so first I'm going to focus on the class 2 peroxidases. So this is a slide now. It's the same tree you've seen before. The bar graphs show you the copy numbers, and the shading on the branches are expansions and contractions reconstructed with a program called Calif. Blue is expansion, red is contraction. And I think it's pretty obvious that the white rot lineages are dramatically enriched in these class 2 peroxidases, which is really consistent with the central role for these enzymes in lignin decay. So white rot genomes have 6 to 26 copies of these genomes, of these genes, excuse me. Brown rot have 0 or 1, as does the ectomycorrhizal fungus. So, yes. Okay, next, I'll show you these two families of cases. One is a GH7, which includes cellobiohydrolases. The other is a very interesting gene family that's been called a glycoside hydrolase, but now we think it has an oxidative mechanism for breaking down crystalline cellulose. Notice that both of them have expansions at the base of the agaricomycetes, and now I'm only showing you the agaricomycotina, the group that contains agaricomycetes, but also reductions leading to brown rot lineages, and so the copy numbers are higher in white rot lineages, and that reflects early expansion and then reduction in brown rot and the mycorrhizal lineages in parallel in these gene families. And this is kind of consistent with the idea that brown rot fungi, having evolved some neat non-enzymatic mechanism for initiating cellulose depolymerization, have sort of cast off some of these cases that are otherwise play that role in white rot fungi. Okay, I want to focus even more on class 2 peroxidases now. We're very interested in understanding the precise pattern of changes in copy number in this gene family and the timing of those changes in copy number. And for that purpose, we can use the class 2 peroxidase family. This is now the gene family. Here are the 3 copies in 3 brown rot species, the 1 copy in 1 mycorrhizal species, the litter decayer, and all the rest of these are white rot things. So, just sort of hammering home the point that there's been massive expansion in these white rot lineages. But when did this expansion happen? It's a very hard problem to resolve, actually, because although we have a tree, a lot of the branches are weakly supported, there's ambiguity, and it's a tough problem. So we've used several methods of what's called gene tree species tree reconciliation, which are methods that allow you to take a gene tree, a gene phylogeny, and try and fold it into the species tree to estimate where those changes in copy number have occurred. It's a very difficult set of problems. We use 3 methods that I don't need to describe except to tell you that they have very different principles, very different assumptions. And the results are here. So No-Tongue, Cafe, and Dr. ML, which are the 3 methods that we used, the nodes now are color-coded according to the copy number of those class 2 peroxidases. Gray is completely absent. White is 1 copy. The cool blues are, what are they, 2 to 5 copies, and then so on up to the purple and reds, which are the highest copy numbers. And so there are some class 2 peroxidases in ascomycetes, the sister group to the Basidiomycetes, although for reasons I don't have time to get into, we think these were not ligninolytic. But anyway, they're present in ascomycetes, so unless you assume horizontal transmission, for which we have no evidence, they must have been present in the ancestor of ascomycetes and Basidiomycetes, and then retained in the backbone lineages of the Basidiomycetes, with parallel losses leading to rust fungi, smut fungi, and then these parasitic and pathogenic jelly fungi, the Tremellomycetes. So all of the methods reconstruct that pattern. All of the methods also suggest that the initial diversification occurred in the lineage leading to the Agaricomycetes, the common ancestor of the Agaricomycetes there, although they differ in the magnitude of that initial expansion. And then they all suggest that there were parallel expansions leading to white rot fungi and parallel contractions leading to the brown rot fungi. So a pretty interesting pattern there. Now, if this is a little hard to stare at, this is an alternative graphic which shows you the same results. These are the phylogenies now just stretched out with the node heights corresponding to the copy number. So it shows you the initial expansion leading to Agaricomycetes, and then the parallel contractions to the brown rot fungi, parallel expansions to multiple lineages of white rot fungi. Okay, so conclusions for the first question. What are the mechanisms of these transitions? Well, white rot fungi are obviously enriched in these diverse decay-related cases and oxidoreductases. Class II peroxidases began to diversify in the lineage leading to the ancestor of the Agaricomycetes, which we think, therefore, was probably a white rot fungus because it contained multiple class II peroxidases. Those peroxidases have expanded in parallel white rot lineages and contracted in parallel brown rot lineages. And cases, the ones I focused on, have also diversified early in the evolution of the group and contracted in brown rot lineages. So a real parallel divergence of modes of decay in these functional classes of fungi. And then finally, we'll address this question of when white rot evolved. And we'll specifically ask, were lignin-degrading organisms rare or absent in the Paleozoic, as Jennifer Robinson suggested? So now we walk out on the thin ice of molecular clock dating. And we've used 3 different fossils to calibrate our molecular clocks. We used one of our data sets used for phylogenetics, a 26-gene, 10,000 or 13,000 amino acid matrix, 2 different kinds of analyses. And in one analysis, we sequentially removed one or the other of these calibration points to see if our results were sensitive to any one of those fossils, because misidentification of fossils can have a huge impact on clock analyses. And this is a result here. This is the same tree you've been seeing with the same color coding for the nodes. Now this is a chronogram. The bars are the 95% highest posterior density intervals for the node height age, or confidence intervals, if you wish. They're quite broad, okay? So there's definitely a lot of error in this. What we're really interested in, of course, is the origin of the Agaricomycetes and that first ancestor that we think might have had multiple class II peroxidases and been a white rot fungus. And we're happy that all of our analyses give us a mean age that's around 290 million years with a lot of slop in the error on both sides. So clock analyses always are subject to interpretation, but we feel reasonably good about these results. So how do they compare to fossils? There are a few fossils I want to mention that we did not use as calibration points, so they're sort of a sanity check on our results here. This is a Basidiomycete fossil from the Pennsylvanian, the Carboniferous, and our analysis are consistent with that, with our Basidiomycetes having been in existence at that time, as also with some septate hyphae that could be Basidiomycetes or Ascomycetes that are even older. And then importantly, these are the oldest sort of definitive white rot fossils, and they are from the Permian and the Triassic, which is later than and therefore consistent with our estimate for the age of origin of the Agaricomycetes. And it fits in fairly neatly with Jennifer Robinson's hypothesis. So this is the mean age of origin of the Agaricomycetes according to our analyses. Of course, the error bars are ginormous, but nonetheless, I think this is very consistent with the idea that at the time that carbon sequestration was crashing, that was when white rot fungi were perhaps diversifying. So when did white rot evolve? The initial diversification of class II peroxidases in this clade began by around 290 million years ago. And so lignin-degrading organisms were absent, not just rare, for much of the Paleozoic. And that implies that the reason there is coal is because there were not fungi that could degrade lignin, right? It's not just that geological processes created conditions that inhibited this ongoing natural abiotic decay process. There's a biotic component. We're not trying, plant architecture, shifts in lignin utilization in plant architecture may have been a factor. The paleogeographic factors may also have been important. We're not trying to negate any of that. But I'd say our results are consistent with the idea that this sharp decline in organic carbon burial at the end of the Permo-Carboniferous and the consequent rise in atmospheric oxygen in the Triassic may have been caused, at least in part, by the evolution of white rot fungi. So if I've been even moderately successful here, the next time you see a picture like this, you're not going to think, oh, cool, dinosaurs. You're going to think, where are the mushrooms? Because surely this picture from the Triassic, we know that there were lignin-degrading fungi present and others. They were having a big impact on the functioning of that ecosystem as well as contemporary ecosystems. So if we want to understand the world, we do need to pay attention to fungi. And with that, many people to thank, members of my lab that have helped, including a group of undergraduates who did a lot of annotation. And they got their training in a class that used microbial annotation tools from here for teaching purposes. And then they came to work in my lab to work on fungi, which was great. My co-PIs on the SAP project, Dan Cullen, Dan Eastwood, Igor Grigoriev, Francis Martin, and Antonio Pizzabarro. A wonderful team at JGI that's been just great, and many collaborators that are helping us make sense of all these data. Thank you. Questions?
Thank you, David. I thought when you said how mushrooms change the world, coming from Berkeley, I thought you were going to go another way. But any questions for David? I have one. The class 2 peroxidases that sort of were the start, you described them as kind of being one of the critical new gene families at the base of these fungi. Is there some sense of what the precursor to that might have been? Is there a structural similarity to some other enzyme that did something else?
Angel Martinez is way more qualified than I am to tackle this, but let me just try. So these are in this plant peroxidase superfamily. There are non-ligninolytic members of these class 2 peroxidases that lack the residues involved in binding manganese and in attacking lignin directly. We're calling those generic peroxidases. That looks like it's the primitive or plesiomorphic condition for the whole group. What their function is, I think, is not well understood. Angel, do you have a comment on that? It's okay? Good.
Thanks. Any other questions? Thank you very much, David.
Thank you.